This is Conceptual Chemistry, Chapter 13, Section 4. So in this section, we're going to learn how proteins are polymers of amino acids. So proteins are large polymeric biomolecules made of monomer units called amino acids. Amino acids have an amine group and a carboxylic acid bonded to the same carbon. So you can see that in the drawing over here. Here's the amine group and here's the carboxylic acid group, and they're both connected to this carbon here in the middle. All proteins are made from some number and combination of 20 different amino acid blocks. If you turn to page 413 in your book, you can see the 20 amino acid blocks that all of our proteins are made from. The side groups are what distinguish the amino acids from each other, and that's what's represented here by an R. So that R just means it's some representative unit. Where did I go? It's not on the next slide yet. Amino acids are linked. It's not going to the slide. Hold on. There we go. Okay. There we go. Amino acids that are linked by peptide bonds that are formed in a condensation reaction between the carboxylic end of one amino acid and the amine end of another. So if you look on this diagram here, you can see two different amino acids here, and the carboxylic acid group of one and the amine group of another react together. They kick off water, so it's a condensation reaction, because remember, when you have condensation reactions, that's when you kick off a small molecule. Water is the most typical. And then you can see how the carbon that's double bonded to oxygen is right here, the carbon double bonded to oxygen, and this nitrogen is right there, and that's where they're bonded together. So this is called a peptide bond. So a collection of amino acids that is linked in this way is called a peptide. The number of amino acids in a peptide is indicated by the prefix. So if it's called a dipeptide, like this one here, that means that there are two amino acids that are combined. If it's a tripeptide, that means there are three. If it's a tetrapeptide, that means it's four. If there are 10 or more amino acids combined in a chain, then it's called a polypeptide. So you can see a polypeptide here at the bottom of the screen. Proteins are naturally occurring polypeptides that have biological functions and have a large number of amino acids. So here's some kind of just a very broad diagram here of proteins. Casein is an example of a protein. It is C1864, H3012, O576, N468, S21. So it's got a huge number of amino acids in it. So plant and animal tissues contain proteins in dissolved and solid forms. Dissolved proteins is in the liquid inside the cells, and it's also in other bodily liquids. Solid proteins are what we use to form our skin, muscles, hair, nails, and animals use to form horns. So the structure of proteins determines the function. And it has 4 levels of structure. So looking at this one, actually we're going to go back to this slide. So the primary structure is the sequence of amino acids in a peptide chain. So it's the order in which they occur, and you can see that up here at the top. The secondary structure describes how short portions of the chain are wrapped in a coil called an alpha helix, which you can see over here, or they're folded into a thin pleated sheet, which you can see over here on the right. So on the left here is the secondary structure that's a coil called an alpha helix. On the right, you can see one that's in the pleated sheet formation. The tertiary structure is the way in which the entire polypeptide chain might either twist into a long fiber or bend into a globular clump. And so this is the tertiary structure here. And you can see this looks like a really long fiber, but then it's all bent up into a clump. The quaternary structure describes how separate protein chains join to form one larger complex. And you can see the quaternary structure here. Each level of structure is determined by the level before it. So the sequence of amino acids actually determines the final shape. In long polypeptides, the number of variations in primary structure is enormous. There are 2 times 10 to the 18th different 20-unit polypeptides that we can obtain from just one set of our 20 basic amino acids. It's because of all the diversity of things that we can form that all these different living organisms are possible. So we can have some slight variations in the functioning proteins and still have it work, but sometimes it can cause a life-threatening condition. Sickle cell anemia is caused by an error of 1 amino acid out of 300. So it's a very small error, but it causes an awful life-threatening condition. So attractions between neighboring amino acids and polypeptide chains are what cause the local contortions that make up the secondary structure of the polypeptide. The secondary structure takes alpha helix form when simpler amino acids are grouped together in a chain. The shape of the helix is maintained by hydrogen bonds between successive turns of the spiral. And let's see, you can see that here. You can see this is a hydrogen bond here. And then there's a hydrogen bond there. So those are the hydrogen bonds that help to maintain the spiral shape. Proteins that have alpha helices can be stretched because of their spring-like structure. Anytime you have something that's all coily, that generally tends to lend to stretchiness. The pleated sheet version forms when you have mostly nonpolar amino acids that are grouped together. And proteins with pleated sheets are strong and flexible, but they are not easy to stretch. The secondary structure can vary from one part of a chain to another. So one part can be alpha helices, while another part is the pleated fold. And I think you can see that in this picture. So you can see how we've got the coil here, but we've got the pleats there. So it can vary from one part of the chain to another, depending on what amino acids are linked together in that part of the chain. The tertiary structure refers to the shape of the entire chain. That's maintained by electrical attractions between amino acid side groups. Factors that affect tertiary structure are disulfide bonds, which I will show you right here. So disulfide, that di means 2, so it's 2 sulfurs. So we have 2 sulfurs hooking together and acting kind of like a bridge or glue between these different sections of the chain. They can also be held together by ionic bonds, and you can see that right here. Ionic bonds are also called a salt bridge, and by hydrogen bonds, and you can see the hydrogen bonds up here. Because hydrogen bonds between side groups often exclude water, they are called hydrophobic. So this yellow area says hydrophobic. Hydrophobic means phobic means like scared of. So this would be like scared of water. So a hydrophobic part of it repels water, it resists water. Hydrophilic connections, and those are the green areas. Hydrophilic attractions between proteins and the aqueous medium that they're in help to maintain the tertiary structure fold. They fold so that the nonpolar side groups are on the inside and the polar groups are on the outside to interact with water, because remember, water is polar. So water really only likes to interact with other things that are polar. Remember, like dissolves like. To dissolve something polar, you need to put it in something polar. To dissolve something that's nonpolar, you need to put it in something that's nonpolar. Okay. So proteins that are made primarily of glycine and alanine tend to form alpha helices, and these align to create long fibers. The tertiary structure of these is called fibrous. Oh, we're not there yet. Well, we'll be there in a second. Keratin is a fibrous protein that is the main component of hair and fingernails, which is this slide. The strength of the fibers is determined by the number of cross links. And those cross links we're talking about are disulfide bonds. Thick hair has more disulfide links than thin hair does. Uh, Disulfide cross links enable our hair to hold a particular shape. So here's her natural hair. You can see the disulfide bonds. Permanent waves can modify the curl in hair. First, you treat it with a reducing agent to break this disulfide bonds. Then breaking the links allows the keratin to become more flexible. Then we set it into the desired shape. And so you can see the hair is being reshaped by curlers. Then we apply an oxidizing agent to restore the disulfide links. You can see the disulfide links are hooked together again down here. So proteins are functional only under very specific conditions, such as particular temperature and pH. Changes in these conditions, so if you change the temperature or you change the pH or both, will disrupt the chemical attractions in the protein and cause a loss of structure. When it loses structure, that means it loses its biological function. A protein that has lost its structure is called denatured. So now we move on to enzymes. Enzymes are proteins that catalyze or speed up biochemical reactions. Enzymes have receptor sites that only allow specific reactant molecules called substrates to fit inside of them. Molecular attractions hold the substrate molecule in the receptor site in an optimal position for reacting. After the reaction, the substrate molecule is released to another, so another substrate molecule can fit in. Some enzymes catalyze the splitting of substrate molecules, while others catalyze the joining of molecules. So some are set to split them apart and some are set to join them together. One single enzyme molecule can act on thousands or even millions of substrate molecules every second. Biochemical reactions couldn't occur fast enough to support life without enzymes, because enzymes are what speed it up enough that it can actually happen quickly enough. So inhibitors are things that interfere with an enzyme's activity. An inhibitor would bind in the place of a substrate. These are important for the regulation of cell metabolism. Once an enzyme has produced enough product molecule, it begins to shut down because the product molecules start to act as inhibitors. When the product concentration decreases, the enzyme starts catalyzing again. So let's just quickly kind of go through the sheets. Going back to the beginning. Where are we? Okay, so proteins are large polymeric biomolecules made of monomer units called amino acids. Amino acids have an amine group and a carboxylic acid bonded to the same carbon. All proteins are made from some number and combination of 20 different amino acid blocks. The side groups are what distinguish amino acids from each other. Amino acids are linked by peptide bonds. Where am I? Amino acids are linked by peptide bonds that are formed in a condensation reaction between the carboxylic acid end of one amino acid and the amine end of another. A collection of amino acids linked in this way is called a peptide. The number of aminos in a peptide is indicated by the prefix. Polypeptide means there are 10 or more amino acids. Proteins are naturally occurring proteins are naturally occurring polypeptides that have biological functions and have large numbers of amino acids. The structure of the proteins determines their function. There are 4 levels of structure. Back here. There are 4 levels of structure, and each level is determined by the level before it. So the sequence of amino acids is what determines the shape. The primary structure is the sequence of amino acids in a peptide chain. The secondary structure describes how short portions of the chain are wrapped in a coil called an alpha helix or folded into a thin pleated sheet. The tertiary structure is the way in which the entire polypeptide chain may either twist into a long fiber or bend into a globular clump. The quaternary structure describes how separate protein chains join to form one larger complex.